I'm Heena Modi of Plant Shift, and this is Alpesh Patel. He works for Dr. Hadwen Trust. We're just going to talk about some of the DHT funded projects, and DHT stands for Dr. Hadwen Trust. So, Alpesh, can you talk to me about some of the projects that you have going?
Yeah, so all of our projects that we fund, firstly, they aim to understand human health and human disease states, and also they aim to replace the use of animals in medical research. So all of our projects have these two aims, that not only does the research take place without any animals, but also the aim is to replace current animal use in biomedical research, but also they have to be scientifically valid. They have to be able to help us understand human disease. So we have lots and lots of projects throughout the UK. It's not one project just in London or up in the north or wherever. We have projects that range from cell culture, which is growing up human cells or human tissue in a petri dish so we can understand what's going on there. We also have projects that are looking at brain imaging, so that's understanding how the human brain works.
And that would use expensive machinery, I'm assuming.
It's very expensive, very fancy machines, but what they enable us to do is actually see how a human brain works, how the human brain is interconnected, how different parts of the brain relate, how different parts of the brain go wrong in different diseases. So that when we look at, for example, something like epilepsy or schizophrenia, some of these diseases animals don't get. Animals don't naturally get schizophrenia. So to look at schizophrenia in an animal model is always going to be flawed. There's always going to be problems with that. So by looking at how the brain is affected in humans using these big, very fancy machines, will enable us to directly relate to the problems that we can see, and then in the future look at how we can go and cure them, treat them, or manage those illnesses.
How would you do that? Would you look at healthy human brains and also look at human brains that are where the human is experiencing schizophrenia or epilepsy, or
so
compare the two?
Yes, so excuse me, the project that we have up at the Aston Brain Centre, that's looking at the magnetic signals that you find in our brains. What they do is they look for these signals, and you can see that in a healthy person you get a certain type of signal and certain areas of the brain light up. Then you look at patients, and they use the machine for patients as well, who are also going on for surgery as well, and seeing what parts of the brain have been affected, what parts have a problem, have an issue, and then they can really look at the problem areas and see how these things have changed and better understand what's actually going on in these diseases.
Okay.
So that's a project we've been funding for a long time. It's a very long-standing project. There's been a lot of really great work that's come out of there. Like I say, the machine that they're using, it's called an MEG machine, a magnetoencephalography machine. It's really at the cutting edge of really understanding and looking at the human brain and how the human brain works. And the other good thing about the machines up there and the work that's taking place up there is they have one of the very first pediatric scanners. So actually looking at children's brains and babies' brains and things like that. And again, rather than having to do an animal experiment or trying to interpret what's happening in humans down to in adults down to children, it's looking directly at children and childhood diseases and seeing how we can better understand what's going on there as well.
Okay. What other kinds of projects is Dr. Hadwen Trust involved in?
So again, a lot of our projects are based around growing and understanding human cells and human tissues. So we have several projects that are developing lots of different models for understanding human diseases in various different organs of the body. And they all form part of a bigger picture, and it's very heartening for us to see that a little bit of a project up at Glasgow that's looking at diabetes, it's growing up stem cells, human stem cells, so to investigate diabetes. That has a link with one of our projects down in London at Queen Mary's, looking at wound healing. That also has a diabetic link as well. We have projects that in the past have looked at pregnancy and things like that. And the scope of work for cell culture is huge, and there are research groups doing all the on a chip stuff, the brain on a chip, the lung on a chip, the heart on a chip, and trying to reproduce very small bits. We're not saying they're growing a whole lung, a whole human lung, but they take what is the essence of the human lung, the smallest unit of the lung, and trying to replicate that on a little petri dish that they can then do more and more experiments on. You can grow these things up over and over again, and instead of having to do these experiments on 10, 20, 30, 40, 100 animals, you can do it on 10, 20, 30, 40, 100 little individual human lungs on a chip to see if the drug has an effect, to see if there's a genetic condition that they can solve through gene therapy or something like that. So,
where does the chip bit come into play?
The chip it's just a phrase for the little bit of plastic or glass that the equipment sits on. So it's called a chip because when you look at it under a microscope, there are tubes going in and valves going in, and there's little circuitry and things can flow. So that's where the on a chip bit comes from. It's not necessarily a computer chip, but it's that sort of scale. So if you look at a computer chip, you've got all the wires and everything and all the circuits going in. It's very similar to that, which is why it's called on a chip. And the other thing is what a lot of companies and researchers are out trying to do is then try and fit all these bits together. So you take your heart on a chip and your lung on a chip and your kidney on a chip, and you put them together and you try and get a human body on a chip. So all the bits are interconnected.
Wow.
So it's not saying we're taking the human out of it, because everything in the system is human based. They're all human cells. It's a bit of human lung, it's a bit of human tissue or kidney or skin or whatever it is, but then you interconnect them, and then you can start asking even more complex questions, like if I add this drug, not only how does it come into the body without having to experiment on a human, because drug testing is inherently dangerous, but I can add it to these systems and say, well, yes, this is how it goes in, this is how the liver then deals with it, this is how the kidneys then get rid of it, and all these sorts of things. So that cell culture and 3D cell culture is a big part of the work that we fund. We also fund computer modelling, computer simulations. So taking data that we already have, human data that we already haven't seen, can we then extrapolate, interpret that information so that you don't have to do any sort of experiment. So we know how these drugs work, we know how these chemicals work, we know how, these are the effects on humans. So can we develop the computer programs and the computer simulations so that you can just, and I'm being very simple, but you can just plug it into the computer, type in some numbers, and out pops, this drug is dangerous, this drug isn't.
Right.
And completely removing the need to try and screen these drugs in animals.
And that's being developed, or that's in existence somewhere?
Both.
Okay.
There are some very basic systems out there that we have funded projects in the past that have developed what are called population dynamic studies that take whole populations and look at how these things work, and people are also developing what are called QSAR studies, quantitative structure relationship studies. They're called QSAR studies. We funded work on that is doing that, is saying if we look at these chemicals, these potential drugs, can we identify, is this going to be dangerous to a human? Is this going to be not dangerous to a human? And there are computer systems being developed at the moment that are looking at that, so completely removing the need for the animal data and saying, knowing human data, knowing how the human reacts to certain chemicals, certain drugs, can we put that into the computer system and predict what's going on? Because again, there is a huge amount of work in terms of regulatory toxicology, and regulatory toxicology is all about, is this drug safe?
Right.
Now, currently, to look at whether a drug is safe, the law requires that you have to do animal studies. You have to prove that it's safe for humans by putting it in an animal. And there are flaws with that system. We all know of all the trials that have gone wrong. We know that the animal model is flawed in terms of trying to work out whether a drug is safe or not. So with all the chemicals that are out there, all the thousands and thousands of chemicals out there that need to be tested, you would need thousands upon thousands, millions of animals potentially to test all of them. It's time consuming, it's labour intensive, it takes a long time. We can't wait 10 years test the chemical if people are going to be exposed to it now. So there is not DHT funded work, Dr. Hadwen Trust funded work, but there is in the field of non-animal replacement technologies, there is a huge push for trying to do these automated, computer-based human cell culture, looking at the cell culture, looking at the 3D computer modelling, looking at all of that together to come up with a very efficient system to say that this chemical, this drug is harmful to humans or isn't harmful to humans.
Alpesh, can you tell me a bit about how scientists can get involved with any of these projects?
Sure. Well, the way we work is we have a call for funding. We put out an announcement to say that we have money available.
Right.
Scientists can then apply to us for funding. At the moment, we have two main calls. In the early part of the year, in the springtime, we have what we call our summer studentships, and what those are a chance for undergraduate students, so scientists or students who are just starting their scientific career, who want to get exposure to the world of replacement science, replacement research, non-animal based methods of working in a lab. They can approach a senior scientist in their university where they're studying, who can then apply to us for some money for that student to spend up to 2 months in the lab doing non-animal replacement research. That's something that has a practical benefit. So not only does the student get to understand how they can conduct scientific research without animals, the senior scientist also can influence the younger generation, the upcoming generation of scientists to say, we can do research without the need of animals, but also from the Dr. Hadwen Trust point of view, it embeds that mentality of not using the animals right from the start, right to these students that are going to become the next generation of Stephen Hawking and the famous scientists and the pioneers in the field, and letting them really start that off. So that's the first way they can get involved. The other way they can do it is very similar, but it's called the Alan and Kathy Stross Summer Fellowship. And what that scheme is for students who have just finished a degree and who will be moving on to a second degree, either a master's or a PhD in a scientific field, and they want to get more laboratory experience during that time where they've just finished their first degree and they're going to start their second degree. And that's for 3 months. And what that does is it's a little bit more than the summer studentships, because they're expected to know more, they're expected to be a little bit more independent, but along the same lines that it's allowing them to start working towards doing non-animal research. It's to give them a chance to say, I can do science, I can make scientific discoveries without having to use animals, without having to inflict pain, just so we can better understand human disease. So those are funding schemes that we run for students, for undergraduates, but like I say, the scientists can apply to us for that. That information is all available on our website, and the days and the times and the deadlines are all on our website. Our other major funding call is in the autumn time, around September, October time, which is our full call. And what our full call is the big projects, the big 3-year funding projects for PhDs, for postdoctoral research assistants or fellows. And those are the ones where people have 3 years to run a project. Now for the PhDs, it's again for early career scientists who want to get a PhD, who want to become a doctor of science. For everything else, it's for those guys that want to develop these technologies and take the development and the discovery of replacement to the next level.
Wow.
So those are our main funding opportunities that we have.
Excellent. So if anyone wanted to find out how to make a donation to Dr. Hadwen Trust, would that all be available on the website?
All of that information is available on our website. It's www.drhadwentrust.org. That's drhadwentrust.org. All the information is there on how to apply for grants, how to what research we fund, what our current portfolio is, so all the projects we're currently running, how to get more information about supporting the Dr. Hadwen Trust, how to give us a donation so that they can fund a specific piece of research if they so want. All that information is on our website.
Perfect. Well, thank you very much for talking to me today.
That's okay, no problem.
Good luck.
Cheers.